Hello everyone. Today, I'm happy to share our research titled Towards Optimized Growth and Taxonomic Identification with Single-Cell Raman Spectroscopy Data for Environmental Applications. Single-cell Raman spectroscopy, or SCRS, is an emerging tool that has been used by a lot of microbial studies. It is cultivation-free and non-invasive, meaning that it is very suitable for environmental applications because no such isolates of related strains were required before such investigation. And SCRS-based taxonomy identification is often a key in those applications. However, there are some knowledge gaps still. First, the machine learning algorithms, including dimensionality reduction, or DR, and the classifiers are selected per lab preferences rather than being standardized. And secondly, biological variances have been proven to impact the SCRS-based taxonomy identification accuracy, but such impact is not quantified. Such variances include cell growth status, for example, and thirdly, it is unclear if spectra from lab-cultured isolates can be directly used as the references to classify environmental cells, despite their different growth conditions of the lab culture and environmental cells. To address these concerns, our study benchmarked 5 dimensionality reduction methods and 10 classifiers under the metric of SCRS-based taxonomy identification accuracy. A 17,000 spectra dual label data set was used for these benchmark purposes. It includes a taxonomy label, including 36 strains, and also a growth stage label, which includes 4 growth conditions that the cells are sampled. This data set is the largest of this kind at the time of this study. Additionally, another 400 spectra dataset was also used for benchmarking the purposes to show that if SCRS-based taxonomy classification of environmental samples can directly use lab-cultured cells as the references. It includes the spectra from 2 genera known as Commomonas and Tetrasphaera. On the bottom side, there is a list of the abbreviations of the algorithms that are used in this study. We first benchmarked the DR and classifier algorithms in this section of the study, and the cell growth stage labels are ignored here. We had 2 major observations here. First, subfig A shows that the supervised DRs in general outperformed unsupervised DRs. This indicates that the supervised DRs can be more suitable for SCRS-based taxonomy classification, and subfig C shows a case study that showing the potential reason that why those supervised DRs lead to higher accuracy. And second, we observed that simple and traditional classifiers can achieve compatible accuracy with their complex counterpart algorithms. This observation suggests that simple classifiers can suffice the SCRS-based taxonomy classification. We then benchmarked the impact of cell growth status to SCRS-based taxonomy identification. We found that not knowing the cell growth information did not cripple the taxonomy identification, though this can be interpreted from 2 aspects. First, high accuracy can still be achieved without knowing the growth status information, and in this case study, above 94%. And secondly, when the growth status information is known, even higher accuracy can be achieved, as high as 99.2%. Also, in addition, beginning of the stationary phase, labeled as S1 here, is observed as the optimal status for SCRS-based taxonomic identification. We also tested the simultaneous classification of growth status and the taxonomy for the applications where those growth stage information is necessary. This is achieved by a 2-step model ensemble, with growth stage called the G step done first, then the taxonomy called the T step done on the next. We found that the GT model in this tested in this study generally had a lower accuracy in comparison to the taxonomy-only classification, which is marked as T only here. After investigation, we found that this could be caused by the propagation of the error from the G step, which only had accuracy of 93.3%, and future work might be needed to improve this accuracy. Nonetheless, the S1 stage was still found as the optimal stage in the GT model. Finally, we tested if lab-cultivated pill cultures can be used as references for classifying environmental cells. 2 lab pill cultures, Comamonas testosterone and Testosphaera elongata, were taken as references to classify the cells from the same genera and identified by fish. The results are promising. Subfig B shows that an 85% accuracy can be achieved. In addition, we combined the 2 pill cultures references with the other 17,000 spectra database, and we retrained the model and classified the same environmental cells. The results are showing in subfig C as a 79% accuracy. This means that extending the SCRS pill culture reference databases could be simple and easy. As one warranted future work of this study, we can extract the important biomarker features from the dataset, including both taxonomy-related features and growth status-related features. We hope that using such information can enlighten more detailed knowledge of cell metabolism and can yield better accuracy in both taxonomy and cell growth stage classification. Here are some key conclusions of this study. First, we showed that supervised DRs and simple linear classifiers can suffice SCRS-based taxonomy classification. This can be used as a general guidance for algorithm choice. Second, not knowing cell growth status does not hamper SCRS-based taxonomy classification, so knowing this information can benefit even higher accuracy. Third, simultaneous growth stage and taxonomy classification is possible. And fourth, lab-cultivated pill cultures can be used directly as references to classify environmental cells, despite their different growth conditions. The authors thank the funding agencies that financially supported this study, and thanks for watching.